Those are the push cores, push core numbers.
Right. So because there's a bit of a delay by the time we see it. So,
Oh yeah,
but that's pretty great. Yeah. Because it's now just showing up.
So that all pops up on the main HD screens that the pilots look at, so they can see
Okay.
what you're looking at.
So now we know what clavularia is. Thank you.
Is it something that
it's an encrusting
wanted
octocoral
for? I see it on the collections list, but only as a pioneer canyon. So I think not for here then.
So Daniel is saying it would be interesting for collection. Is this something that we could snip off, or? And then
yeah, slurps are full, and these things will just retract down to little nubs if they're disturbed.
Okay.
So it's going to be very hard to, I don't know if anyone else has ideas, but.
I mean, we can put corals in with sponges in the slurps, right? Or is that just in the bio boxes?
That I do not know. That's a science question.
I'm not too sure on that. Yeah, it's something that we'd want to check with the lead scientist.
Okay.
That'd be okay. In terms of mechanically, I don't think that's an issue.
Yeah, okay. Well, they're saying no worries then right now since
yeah, we've seen it before.
We'll likely see them again. So, okay. They're really beautiful. I'm sure we got some great screenshots.
Okay, are we moving on then?
Really pretty shot. Just that you've got framed up right there. It's beautiful. It's so
I'm
colorful,
going to
alive.
Jess is back, so are you back? You ate already? My gosh, did you even taste it? She wants to come in and play, so I'm out of here. Thank you very much.
Thank you. And the view of Hercules from Argus, this rock is also kind of an interesting view to take a look at. If you want to see what this rock looks like from above and how it large is relative to Hercules, you can see that on channel 2.
That's a great view from channel 2.
You're coming in all choppy on V-Link, so we're not catching any of this.
What about now?
That's good.
Now it's great.
Okay.
What'd you do?
I didn't do anything.
Oh, okay.
I just took a pause. It seems to be inconsistent. But I'll try and
Thanks everyone.
speak more
this is
slowly.
Renato signing out.
Thank you, Renato.
Bye. Ciao.
Grazie. Yeah. Arrivederci. This is a nice shot to rest on as the Nautilus crew comes back.
Definitely, I think it's a beautiful shot here. Could we get the lasers turned off just for a second so we could get one without the lasers?
Jake?
Lasers off.
Hello, folks.
Hello. All right.
Hey, so Jess, are you back?
I am back, yes.
Great. How was dinner?
Oh, tasty and very satisfying.
Good. Those of us who are still ashore, we're lamenting that we're missing that part of being on a ship.
Yeah, roger that. So from my understanding, this is not going to be a photomosaic candidate, and we're still going to mosey around.
Yes.
Correct. We were looking for brachiopods on this rock. We didn't find any, but we're going to keep our eyes out for that in this type of habitat periodically as we go along. But yeah, we're still looking for that photomosaic target.
Roger that. All right, and we're just going to follow along this ridgeline there, Ben?
Yeah, I think so. So I'm going to do 300 to keep on this same.
Sorry, can you repeat the bearing?
300.
300. Roger.
Yeah.
Right.
Can I
and
just
so they were
refresh my
using-
memory?
Yeah.
Go ahead.
I just was wondering, I think we're supposed to have ROVs on deck. I don't think it takes long from this step, but what's our bottom time to leave bottom?
Yeah, so we'll be about 30 minutes in the water column, 15 minutes on the shore or on the surface recovering, so probably around 7:15 or so. We'll leave bottom.
Okay. Thank you.
Okay.
All right.
Does
So
that sound
about
good?
about 2 hours left.
Yeah. 2 hours left.
It can go fast.
So if you guys are all clear, then I will kind of pull away from this rock and we can continue on our mosey.
Sounds good.
Roger.
Yeah, and they were using the sonar to find some candidate targets.
Roger.
Roger that.
Yeah, we can keep on the cliff and look for more local features in the sonar.
Right, and there's a beautiful shot of I have to say this because I know what it is, a rose thorn rockfish, a little bit to the left. We're looking for something where it's about 5 meters in height, but has a good mix and relative sizes of sponges to the substrate. So if there's a couple of larger specimens on the rock, that would be best.
Roger.
Instead of a whole bunch of smaller ones like this one.
Roger that.
Thanks, Jess. So do you prefer Jess or Jessica?
Jess is fine. Easier to say.
Okay. Yeah. Just wanted to make sure. The times when I would say Jessica, I would think, oh, maybe she prefers Jess.
Sorry?
No, it's fine.
I
Oh,
wonder
yeah.
if it's like for me, I'm a Jen or a Jennifer, but not a Jenny. Are you not a Jessie?
No, I am not a Jessie.
That one's a no.
Okay, and
Skate
so
ahead.
then what is
Long
that?
nose skate.
Yay!
Yeah.
Male or
Yes.
female?
I think I see claspers, so male. Did you see claspers?
Can we back up? Can we back up so we can look? We can do a little biology lesson.
There was a shark outside, and I am convinced it was the first great white I've ever seen, but Jess was unconvinced.
I don't know my shark biology very well.
It looked like a juvenile or a smaller great white.
And then zoom in a little. Because I think that's a female.
Yeah, our video is just switching out for dinner, so this might not be the best zooms.
Okay.
Yeah,
What do
no.
you think, Jen? Are you still there?
I'm back. My audio got really glitchy, so I came back in. A zoom would help for sure clarify, but
Video,
I
do you
thought I
wanna
saw
do
some
a zoom?
claspers under that tail.
Oh, maybe. Oh, you're right. Maybe that sort of that white along
Mm-hmm.
the edge there. Yeah, those are claspers. Very good, Ben.
There we go.
Sorry, Jess, did you ask for a zoom?
Ooh.
Yeah, but it's all good. I think they've identified it now.
Okay.
Yeah, the other ones we were seeing,
But
they
there's-
weren't like totally on the ventral side. They were kind of spread out, but
But
who knows?
they are really cool looking animals, aren't they? Just really
Yeah.
beautiful.
All right, Marley, come wide, please.
All right.
So 300.
Yep, I'm going to call it a no.
Thank you.
Roger that.
I don't think we've yet seen any of their egg cases around, but on a past dive,
on
as I said earlier. I can't
previous
remember.
watches. Yeah.
Oh,
we've
you
seen
did?
them. Yeah.
Okay. I didn't catch that, but there was Christmas tree coral at one point that had sponge on it, and then on the sponge were
And then it
I
looks
think
like
cat shark
you're
egg
pointing pretty
cases.
down at
That was
me,
really
so I'll
neat.
get ahead of you, but it looks like there's some rocky candidates to the right.
Yeah.
Sorry, Kim?
And for viewers at home, if you didn't know, the video from dives is posted to their YouTube channel, so you can always go back and look at past dives. To the left, what fish is that? I think it might be a ratfish. Can we pan left?
Sure.
It may have already skedaddled, but did you see that fish that was swimming with its pectoral fins?
I
No.
didn't see it.
Oh, where did it go? Oh,
Oh, I
there,
can
just
see
a
it.
little further to the
You see
left.
the
I can see
shadow
the
of it.
shadow on the
Shadow.
seafloor.
Yeah.
The shadow nose.
Oh, chimera.
Oh, nice.
Ah, ha, ha, yes, they're so
Oh, yeah.
cool.
Lots of white spots
Let's
on
zoom
this
in on
one.
this.
Oh, stand by,
The
Marley.
males have
Great.
an appendage on their forehead, which is really strange and interesting.
All right. Go ahead, Marley. Oh,
So what I, woo,
Little standoff.
yeah. Looks like in front of a coral, no less.
And there's
beautiful.
a hagfish.
All
Just
right.
for you.
Yeah.
Thank you.
Sure.
I think ratfish are really interesting animals. Also referred to as chimera. They're a cartilaginous, a fish with a cartilaginous skeleton instead of a bony skeleton. So they're in that way more similar to the skates and rays and sharks.
And they're sort of ancient, right? They've been been around for a long time. And
Chondrichthyes, yeah. But
yeah.
everything's ancient. That's always an interesting thing to me. It's like we're all just as evolved as
Well,
everything else.
Well, yeah, it's like which time scales are
Right.
we talking on?
But they
molas
branched
are
off
more
from the
recent.
bony fish way early. So
Yes. So if you think about mulas are the most derived or the most recent
speciation.
fish compared to they haven't been around as long as the chimerids. But yes, compared to us, everything's ancient.
When I was doing my research for my dissertation in Monterey Bay, I was catching little flatfish, which is when I became quite the fan of the flatfish. But one time, unexpectedly, in my little trawl net, I caught a ratfish, and it was a male. They have these spines on their foreheads, and it kind of had attached itself to the net. It was very hard to remove and extract it. We were able to do that, but it was very unexpected since we were in about 60 feet of water to catch something I expect to be seeing in much deeper waters. It was really neat to get to see it up close.
So what we just saw was a ratfish, which aren't the same as rat tails, which are the grenadiers, which we saw a lot of at the more deeper depths. They have sort of similar shape.
They swim very differently. I mean,
Yeah.
what I keyed in
Different.
on is these other fish that we're seeing are swimming in a different way. They're not using those pectoral fins, the paired fins that project from the side of the fish. And you may have noticed that that was the predominant way that fish was moving. When it was kind of cruising along, it can still use its tail for more rapid propulsion. But when you're looking at fish sometimes, the way that what fins they're using to move can help you figure out who they are.
So some of the I can see it now. That was a black gill that we just passed because you could see the black spot on the operculum. Now that I've seen it, I think I can hopefully see it again. So that was one that we just
Is
passed.
that a line?
Some beneath us?
Right. Little fishing line.
Mm,
Look at that.
Can't really tell.
Whatever it is, it's very sedimented.
Yeah, it's been settled for a while. I don't see it continuing.
Yeah.
Okay.
That's good.
sounds good.
Good eyes.
Well, seeing as you're much more proficient at looking at the sonar than we are, do you see any promising targets coming up?
Not in the
None
immediate
of these
future.
Okay. Lisa, while we're looking, should we do another transect just to make sure we get that last one in?
Yes. I was thinking the exact same thing. Although you and I have
Okay.
not worked with each other for a while, we still think alike.
Sure, would you like to start your transect now then?
Sure.
All right. And you want to drop a waypoint, then I can just continue
Yeah.
on 300?
Yeah, we can continue on 300. It looks like there's a slight bend, but
Sure, we can adjust.
yeah. This will be transect 12. Is that correct?
Yes, correct. Roger that.
Roger.
What does that come from, saying Roger?
I don't know.
I don't know.
Maybe there's a Captain Roger.
All right.
Or it comes from
Pushing
Righto
forward now.
Roger. I don't know.
All good there?
Yeah, I'm okay.
Lasers on?
The ship's moving, so.
Yep, roger that. Yes, lasers are on.
That'll be my next Wikipedia search. Roger etymology.
I'm seeing a lot more urchins right now in this little area. They're the little pink spheres on the seafloor that look like pin cushions. And we've just started a transect, which means for the next 200 meters, we're going to be proceeding at a fixed distance from the seafloor at a pretty constant speed, so that later someone can come back to this video and measure and count the sponges and corals, fish, etc. that we're seeing as we're transiting along the seafloor. Anything else we should add to that, Lisa?
That sounds good. And I noticed that Di saved us from, or saved Ben from doing a Google search. It comes from Roger Wilco. Roger Wilco, procedure words for radio telephone communication, meaning received and will comply. I guess we got rid of the will comply part.
But we will comply. For sure.
What's the one of the easiest to distinguish words?
Oh, I think you're breaking up there a bit.
Yeah, unfortunately, Jen.
Okay, how about now? So that,
It's still garbled.
yeah.
Yeah, it might be best if she can restart V-Link.
So restarting really means that we need to restart our computers, because I don't know any other way. We can't really Or maybe somebody can explain it to
Well,
me.
yeah, just exiting out of the application and then often a full computer restart. Usually,
Yeah.
I know it's a little bit of a pain. It can be a bit inconvenient.
Yeah,
but
because, yeah, as you know, we all have
Marley.
our computers set up and.
yeah. Who just said
Are you
Marley?
suggesting, Jennifer? So I did the application restart, but are you suggesting, seems that Lisa's audio is functioning better than mine, and she did a full restart of her computer, and I only did the application restart. Should I go ahead and do the whole
Well, you sound fine
restart?
now.
Yeah,
It's
you
not
sound
sounding
good.
garbled
Oh.
at all now. So let's just sit
Okay.
tight, and
Okay.
if we start to completely lose you again, then maybe a full system reboot would be good. But I think for now, if it ain't broke, don't fix it. Let's just
Okay.
hang in there.
Sounds good. I don't know if you even heard me laughing, but I laughed because what I was telling you when you couldn't understand me was that Roger is supposed to be one of the clearest and easiest words to distinguish over the radio. And you couldn't hear anything I was saying.
Oh, the
So
irony.
I just found that ironic.
Even Roger didn't get through.
Well, and what you just said is exactly what Kirsten Lindquist sent over the science chat.
Oh, I know.
Verbatim.
That's
Okay.
what I was telling you.
Okay, good. I was like, wow.
Thank you, Kirsten.
Yes.
But it turns out if you don't have a good radio connection, it doesn't matter if you're saying Roger 10 times. No one's going to tell what you're saying. We have a little red coral that just went out of view, a number of other of these purple, whitish pink paragorgid corals or bubblegum corals, and lots of small to medium-sized sponges. Oh, and a flatfish over in the bottom left corner, going out of view. It was hard to get a close look at that.
There's a flatfish under that rock on the right too.
Oh yes, so that one has
Dover?
eyes that are more bulbous. I'm thinking, yeah, Dover. The Dover has eyes that kind of protrude up off the
Seems advantageous
fish's
to have
head
eyes
more
like
than
that.
some. What's that?
Seems advantageous if you're flat like that to have eyes that protrude. Little periscopes.
It can,
Yeah,
yeah.
I
I
can see
mean,
far.
yeah,
Eye stalks.
but they don't all have
Yeah.
that. Only some species do.
Yeah, I wonder what differentiates that functionally.
I mean, now that we're on the topic of the flatfish, we could talk about how fascinating they are. So I have to look at the depth distribution now, but when we were in deeper waters than this, all the species that we were bound to look into are called run into are called right-eyed flatfish. And that has to do, but there are also left-eyed species, and that has to do with which eye migrates. Ooh, what a beautiful migrates over the top
Which
of the
nav?
has to do with which eye moves during metamorphosis. So the reason they look so different than all the other fishes, they start out looking like a normal fish with swimming in the water column, and they have a right side and a left side, and each of those sides has one eye, like we're used to. But then as they start to transition from living in the water column to living on the seafloor, one eye transitions to the other side and joins the other eye on one side of the body. That's the side of the body that develops pigment, and the other side usually stays kind of clear or whitish in color, and that's the side that is facing the sediment, and the pigmented side is facing up. So it's a very kind of different way to live and different way to develop, but it's pretty interesting. And there's videos online where you can actually watch the animal transform in kind of a rapid, speed up video. It's pretty neat. Okay, we've got another skate.
Long nose.
Nailed it.
I think I see claspers.
Yes, and
Well, now-
clappers.
Yeah. The claspers we were seeing
Yep.
before weren't so underneath on the ventral side, so I don't
You're
know.
right.
They don't.
The-
I don't know if they have
these
anal fins too that were confusing. but we have the common name right for sure. Questioning everything after just didn't think that that was a great white outside. It's very exciting to me.
You are in their where you can find them.
Oh yeah?
We are in there.
I'm pretty convinced.
Oh yeah.
I'm pretty convinced. It's fat body and white under shading. I don't think I've ever seen one in the wild.
The juveniles hang out in Southern California, and then as they become older, they move further north to where the Greater Fairlawns area is, where they have a lot of prey items that the adults like to feed on, like the sea lions and the rookeries that are up there, and elephant seals.
So you would get more smaller size class, smaller cohort
Around
here.
Yes.
Yes, it's
down
all
in
lining
Southern
up.
California.
My dream come
Yes.
true.
No. Yeah. Another flatfish. Is that a? Did you see that?
Yeah,
Jen?
just
Where?
out of view on the
I
bottom
missed
right.
it. Oh,
Yeah.
I was looking up
There'll be
what,
more.
well, no, at this depth, yeah, I was looking to see what species we might find. Most of my, time looking at flatfish has been either deeper or shallower than this, so I'm not sure
Oh, and there's one
exactly
right in
which
front of us
species are
now.
this.
Yep, right
Yeah,
front
it
middle.
looks slender and it has the bulbous eye. I mean, I'm gonna guess it's a dover without closer ability to look.
So what's the
Flatfish
common
is gonna
name?
be a little tough. Go ahead.
Yeah, what's the common name of the one that doesn't have protruding eye stalks?
Oh, there are a number of. I'm not sure at this depth.
Right.
But are you just curious to know any of them that wouldn't have the protruding eyes?
Or just
like
that
the
we've
English.
seen today, or
Oh, well, have you seen deep sea sole? Because they're not as obviously protruding their eyes.
And
You look up
Ben,
the
what's the
depth
distance we've come on this
distribution of
transect?
deep sea sole.
yeah, we've come about 130 meters already. Or no, sorry,
Okay.
90 meters.
90, Raj. Looks like we might want to
Apparently,
do
we
a
went by some.
science. Do you want us to stay along that ridge, or do you want us to go direct to AD4? It seems we're going a little off the ridge now. We can go back and kind of do a little bit of a heading change, or we can continue over to AD4. It's up to you.
It won't really affect our time to get to 84 much to go up onto the ridge a little more. If that's...
Yeah, I think that might be beneficial.
Yeah, I think that might be easiest to just stay on this heading.
Okay.
Sure.
And
Thank you.
from what we've seen, we probably have a more likelihood of finding the target we want for the photomosaic at the top of the ridge, it seems.
Might be,
Would you agree?
yeah. It seems a little bit more sedimenty, smaller rubble down here.
Mm-hmm.
So the top of the ridge would be a heading change to get there. So we can do that.
You can do it pretty gradual.
Is that okay, Lisa?
Yeah, that's fine. It gradually would be good.
Okay, so we'll just add 20 degrees, 320, maybe 33.
Yeah, you want to show me on the map?
Yeah, let's do 335 to start. Is that good?
Yeah, 335
Okay.
works. Is that okay with you, Kim? I'll start my lateral now. Roger that.
So we're heading towards the ridge.
Yes, that is correct.
Okay,
We're just
yeah.
going a little bit more
And that-
upslope along that contour a bit better.
Yep. Yeah, there was a little bend in the ridge as we approached that 84. So, yeah.
Roger.
Marley, could you switch out 3 and put in high pack for a moment?
Sure.
So we're going to do a heading change now too. So I see into the slope.
Yeah, and the ridge will come to a local max right above 84 if we want to keep on the ridge until recovery.
Yeah, that sounds great.
Okay, cool. We'll work our way up
Can
there.
you see it now, Lisa, in high pack?
Yes. Thanks for putting that on.
Sure.
Great.
So, I have to say, oh, there's another flatfish. That wasn't what I was going to say, but I had to point that out too. That I've had the pleasure of getting a chance to go out on the Nautilus in past years, and Lisa has not. So she is had to learn how to do all this without ever having the experience of being on board, and I just think that that's pretty great to point out. Good job, Lisa, and you're doing a great job.
Well, you're being very generous, and I have to say that I have benefited from a lot of people helping and being, generous with their time. And I think at sanctuaries, we really know how to collaborate and work with partners. So I think you've noticed that as on our science team, we have lots of people from National Marine Fisheries Service, and it's really great to rely on expertise from others that you don't have to always possess it yourself. And there's no way that you could keep it all together, right? So we really rely on other people.
Science is definitely collaborative.
Yeah. It's just been great having Meredith and Steve Oskovich. I haven't had the pleasure of working with him, but they're coral experts. And Abby is a sponge expert. And I'm not sure on the Nautilus crew, are there other experts that I'm not aware of that that maybe Ben or Jess you could tell us about? Like, what is your area of expertise?
Ben goes first.
Yeah, actually, let me just call on this move.
I think that's him punting to you, Jess.
So my work, I'm in a graduate student. I'm finishing up my PhD, and I'm interested in kind of midwater ecology. So I have a background in plankton and some larval fish ID work, and just kind of looking at using imaging and acoustic in situ via sensor platforms to kind of combine the biological information with the fine scale hydrographic information, which in this vast 3D midwater space, which is the largest habitat on Earth, it's really technologically challenging. So I work in a marine robotics lab trying to kind of apply these novel remote surveying platforms to look at data that couples the biological information with the fine scale environment. And yeah, it's kind of a neat, very much technology enabled field. But there's a lot to find out because from nets and from shipboard acoustics, you can only get so much understanding of the fine scale distribution and how that's interacting with the environment. So that's kind of my interest.
And what area of the ocean are you studying?
Yeah, so the midwaters, anything between kind of the sunlit surface layers and the seafloor, kind of the two interfaces. But the mesopelagic, kind of 300 meters to 1,000 meters, where biology is kind of still affected by the photo conditions of day-night cycles, but there's not room for photosynthesis. That's kind of a major middle box in the ocean and in the Earth system as a whole. So there's a lot of interest in kind of understanding this open ocean pelagic ecology that you know, you can only get a resolution with these kind of technologies that can source information in situ at depth. So yeah, that's my interest is, you know, all these diel migrating layers and these fine scale patches.
Wow.
And, yeah, it's a fun field, but it's kind of emerging with the technology.
That's cool. And Jess?
Yeah, very interesting.
Yeah, so for myself, I'm also doing my PhD in material science and engineering, and I work in a bio-inspired robotics and design lab. My main focus area is on developing reversible adhesives for use underwater, specifically towards manipulation and locomotion capabilities. So yeah, so some of the interesting things is I've been studying certain organisms for how they adhere. They're pretty interesting reversible adhesion capabilities, and then developing kind of artificial analogs and synthetic adhesives for applying to manipulators specifically. I guess underwater and also for surgical applications. So also been playing around with surgical manipulators and applying the adhesives that I make to these laparoscopic tools. But yeah, so taking inspiration, as someone had mentioned earlier, you can learn a lot from what nature does. And they have many years, millions of years of evolution to do a pretty interesting iterative cycle.
Yes.
So
most definitely.
yeah. Probably one of the most famous examples of an adhesive currently used in robotics, in addition to like Velcros and things like this. But there's a type of gecko-inspired adhesive that functions based off of van der Waals forces. And kind of people have been studying gecko toe pads and things like this for a while, but made these microscopic structures. And these microstructures are able to basically allow for the same type of van der Waals adhesion in air.
Van der Waals adhesion in air?
Yeah, so van der Waals for the gecko-inspired adhesives, but there's not as many studies gone on yet into kind of reversible underwater adhesives, and that's kind of the area that my PhD has been taking me for the past 4 or so years. So, yep.
Thank you
Pretty
both
cool.
for sharing that. Yeah. So this is a nice ledge with smaller corals and sponges. I think
we're getting
I
up on
see
that
a
ridge now.
clavularia, some small specimens of clavularia.
Look at that one smaller sponge with the dirty, but really shaggy exterior. It's really interesting.
Yeah.
Tom had a name for those, but I forget. The hairy boots.
Hairy boots?
I think that was one common name.
Who's coming in? Who's this rockfish? I don't see looking at the...
We're definitely coming across the shallower ones. That was a shaggy dog sponge.
Shaggy dog, there we go. So we've come 200 meters since the beginning of the transect, so if you want to mark it.
Okay.
Good. Okay.
So that was the 12th. Is that right, Lisa?
Yes, we're at the end of the 12th. I'm just waiting for the data
Right.
logger to mark the time. That's Mary. Thanks, Mary.
There we go. So now we're on the hunt for something very three-dimensionally interesting. Again, Lizzie reinforced to us in the science chat that we're not married to these waypoints. There's suggestions to try and.
Yeah, so we're kind of approaching the top of the ridge on this bearing, and if we kind of keep up on this ridge, there's a local maximum, a little mound right above that AD4 target. If you want to just keep on the kind of the ridge, that's something I might suggest. You can see in high pack, I don't know, but that's up on the third.
Yes, sorry, in the middle of what I was saying, I cut out.
Sorry.
But oh no, it's not your fault. But yeah, so we don't, we don't have to go to AD4. We can definitely go. I think, like you're saying, go see what's at that higher elevation, and and look around for rocks and ledges that might make for interesting targets. We're also going to continue to keep our eye out for those brachiopods. When we get to ledges, we can zoom in and look a little bit for them.
All right, I think I heard that. It's a little hard, but yeah, we can keep on this ridge if that, and we'll look in the meso for local features that are kind of structurally interesting and maybe overhangs is what you're saying. Okay, great.
Yeah.
Sweet. So.
Yeah, it looks like we're right along that ridge now.
Yeah. Sure.
So maybe looking off of that, maybe we want like a 3-0 or 310 or something like that again.
Yeah, 310 is what I want in the high pack, but yeah, I'll just keep us there.
Yeah. $305, Raj.
Here we go.
305, Kim. Roger that.
And Ben, do you have the boundaries of the marine protected areas in high pack? I know that AD4 is going to be still outside, but the closest, if we crossed over to 85, we would enter into the marine protected area.
I can check in the
state
plan
reserve.
real quick, but I don't think it'll be in this survey you see here.
Okay, it's closer to 84 than 85, so I would say it's kind of near where there's the next black vertical line. And so if we end up over there, we'll just, and we wanted to do collections, we just have to think about that. We probably won't, so I'm just putting that on your radar, though, your mental radar.
Yeah, Roger. Yeah, this ridge is only about 100 meters up from 84, but it is in the direction you're saying would be. Wait, are you saying that 85 is close? To the west?
If we went west, not north, but west of 84.
So we should be.
We would enter the State Marine Reserve, yeah.
Okay, so
Ooh, what are all these little white dots on the rocks? Can we just zoom in case those would be the brachiopods we're looking for?
Sure. Sure thing.
They might be sponges or something, but couldn't hurt to look.
All right, Marley, go ahead and push in.
It's polymastia, the yellow.
Oh, yeah. Yep, yellow. Okay. What about the rock to the right? There were also the larger rock with the yellow sponge. There were some, maybe, just zoom in on this.
I'm going to move in a minute. Marley, you want to do a quick push?
So Daniel, if you're still watching, holler out through Science Chat if you see anything that you think looks like a potential location.
You want me to stay at this rock, Science, or do you want, we have a ship move?
It's okay. I didn't see anything that jumped out at me.
Okay, Roger, we'll continue.
Ship's so responsive at this depth, it's not.
Roger. All right, Ben, you can go ahead and call it back in.
So I think our, I know that our sampling folder has a picture of the brachiopods. If you went to
okay,
our table of contents and then went into the sampling folder.
Okay. Great. I mean, I know in general what brachiopods look like, but if there's some specific about
Yeah,
what we're looking for.
And it's under it's all the Santa Barbara SB, NMH, NOAA, cruise, Malay protocols. I think it's in there. There's some pictures. Oh, no, not that one.
I found it.
You found it?
Thanks.
Good.
Yeah. Looks like there's supposed to be imagery, but they're not loading.
Yeah, so it's a PDF. It has the word
I'm looking at the wrong
references,
thing then.
Images. Yeah,
oh, okay. Now I got it.
So, yeah, so they sort of look like, to me, they look like oysters, but not as irregular, like a clam / oyster, how they're stuck to the rock.
Okay, so yeah. Okay, that's helpful. Thank you for letting me know about that.
Hey Jen, have you seen the MBARI AUV imagery and multi-beam of brachiopod beds up in the bay?
No.
In Monterey Bay?
Yeah, they're pretty cool. These, their AUVs now do photomosaicing and sub-centimeter multi-beam. Obviously very small swaths, but they've come across several brachiopod beds, and so they have the three-dimensional data plus the photomosaicing, so they can build the 3D habitat structures, virtually. But it's pretty amazing how precise and high resolution that tech is getting.
It's a fancy data set. Sounds really cool.
Earlier this year, we were actually scheduled to use MBARI's AUV, not to get the sub-centimeter sampling, but the sub-meter AUV multi-beam out at the Octopus Garden area, and then run photomosaicing.
Can we push in a
AUVs
little
over
closer on the video? Sorry, just wanted to, while we're looking around, if we can get any closer looks on these rocks, that might help with brachiopod sightings.
Yeah, we can zoom a little bit. It just depends on how comfortable Jess is with flying with the cameras.
Well, even we've gotten a little closer to the bottom, so that helps too.
guys want to zoom on something specific here?
Maybe we should just peek at a few of these rocks up close. Is that okay, Lisa? Or
Yeah.
do you want to keep searching for the target?
Well, why don't we just try and find something that's a little bit larger to look under? Because I
Okay.
think that
Okay.
these are very low relief, or
Okay.
low aspects.
Sounds good. I interrupted you, Chad.
Oh, no worries. But COVID, of course, got in the way of that particular mission, so it's been put off indefinitely. But the idea is to take the AUV out and to do sub-meter multi-beam mapping, and then to follow that later with the photomosaicing AUV to try and find these octopus nurseries around rocky outcrops. It's just probably a quicker and more efficient way to find them if they're more plentiful than we originally thought, which they may be.
So, Chad, if we're using ROVs, remotely operated vehicles, why are these called AUVs instead?
Yes, good segue. AUV is autonomous underwater vehicle.
Thank you.
So just think of it as an automated underwater drone. And so scientists, they look kind of like submarines or torpedoes with wings. And they can program them to run specific pathways underwater at certain heights above the seafloor. And you can put different payloads in them in terms of sensors and equipment, like the camera equipment, multi-beam, all sorts of stuff. And so they'll just send them off on the boat, like sending your child to kindergarten, and hopefully you get your kid back at the end of maybe the 2, 3, 7-day mission. So it's pretty cool.
Oh, yeah.
Stuff that no one really thought about, or that could be possible 20 years ago.
Yeah. Did you want to see the octopus, Lisa, or just point it?
No, we've seen lots of octopus. I'm sorry for people who are listening right now, but we've harassed.
Okay.
It's gone now.
We've harassed enough octopus, you're saying, today?
Yes. Well,
Okay.
just period.
There we
They
go.
are definitely charismatic. Oh, this is really pretty here.
Yeah. Just a reminder that the Argus view is really cool.
Yeah, you want to stop the ship then?
Yep.
And the Herc view is really neat, but the Argus view is also pretty interesting when you are looking at features like this.
Yeah, so we can
Okay,
explore along
Daniel.
this ledge here.
Daniel.
Hold on,
The Santa Barbara Natural History Museum is liking this ledge.
Yeah, so look under. Can we look under this ledge?
Mm-hmm.
Yeah, sure.
Thank you.
Also, notice it looks like there's some basket stars on those yellow sponges. Each sponge has its own little basket star on top
No sharing.
of the lid. Only 1 star per sponge, please.
So, Daniel, if you were a betting man, would we find them here?
If anyone's wondering who we're talking to,
Yeah.
need close view for brachiopods, please. Thanks. So we not only have the folks that are on the Nautilus vessel helping to run the ROVs and the science, and then you've got Lisa and I helping from home. Then we have
push on in there, please.
a number of scientists who are also in their homes participating in the science chat as scientists ashore. And so they're a huge support to us and help to make sure that we are doing the best we can to accomplish multiple scientific objectives.
Oh, this is really pretty. And look at the diversity.
Yeah, I see some tunicates and some sponges. A brittle star peeking out from that little crack up there at the top. So he said it could be the kind of habitat where we might find them.
Right.
Pan along, please. So if we can just pan along
Sure thing.
the side of this ledge, that would help looking for them.
Be a little jolty.
That's okay. Take it in steps.
You can shake us all about.
Oh, all right.
There
Well,
was a
that's the end of that
beautiful
rock.
translucent snail.
Okay.
I did not see that.
Are
Oh,
they gonna
there's a
come a little
tiny
wide?
rockfish.
Where?
Right there. It looks like a rockfish to the
Look.
left.
Oh, in the background?
Marley, full wide, please.
Yeah, in the background. Very small.
Look for a place that's well lit, but I'll zoom out so we don't get you guys motion sick. All right, Marley, go ahead.
Are we panning in the other direction now? Do we want to zoom in
Can we-
further?
We can zoom in further to this
Can
one.
we pan right a little more, too?
Sure. Yeah, you're right.
Boing.
The spicy pan and tilt for us here. One more.
Is that purple sponge all the way on the right?
Is that an egg sac down there, or is that a brachiopod below the sponge there?
Oh, the bottom left.
Science, do you see that little dangling?
It's an egg sac.
It's an egg sac. Roger.
Maybe. Like a skates egg sac?
Oh, yeah.
Is that the color you're talking about? Like a
Yeah,
muddy green?
like a yolk in the
Okay,
middle
I
of
think-
a...
Oh, Roger. All right, Marty, you want to come a little wide? I'll pan around. I'll scoot around this guy.
Okay. What is that?
Gastropod on the right? That spotted?
Far to the right. Yeah, it's spotted. Looks like the Death Star from Star
yeah.
Wars.
Let's look at it.
It's very small for the Death
I
Star.
just love our film reference.
I mean, the Star Wars universe, we think it's big. Maybe the Star Wars universe is very small.
Star Wars universe is just in the deep sea. That's a shell.
Well, that was what was so great about the Avatar film, that I felt that so much of it looked like undersea organisms. The way that the whole planet was created. Pandora, I think it was called.
It's been a long
that
time
James
since
Cameron.
I've seen that.
Waiting for a sequel for like 10 years.
Yeah, but I think he took a lot of inspiration from
Marley,
what
push
you
on in.
see in the ocean environment.
That's cool.
Yeah, but you ready for 4 more of them?
No.
No.
Right, maybe
That's what they're
not
making.
a shell. What is that?
Yeah,
What
what
is
is that?
that?
Are we want to zoom in on this guy on the upper part of the screen?
Yeah.
Looks like it's pockmarked.
You want to come a little wide?
Yeah.
It's the gray dotted, sort of gray with
Yeah, we're just trying to
holes,
get the camera
symmetrical
oriented.
holes.
The dots look almost like 3-dimensional, like it's something on it. I don't know though.
Like cat. I think they look like they
Oh, is
are
it
cavities.
osculus?
Cavities.
Is that a sponge?
Like
with
they're
osculus?
depressions.
Oh, it's a sponge.
Yeah. It's
I
neat.
wish somebody were on. We'll just write down unknown.
I'm gonna take a screenshot with my phone. I'm gonna harass Amanda with it.
Zoom and enhance.
Yeah, that's full zoom.
Really
Be like,
here.
what is this? This is how you lure Amanda into the science
That
chat.
is an unusual one, right?
Yes, Sponge, please collect if we have space.
Yeah.
Oh,
Is what Kirk
ooh.
says.
Ah.
Do we have space?
Mary,
Ooh.
do we have
Abby would
space?
be interested.
If you are okay with